We are now on part 2 of the chapter 7 recordings, and we're going to talk about passive transport, which is movement of a substance across a membrane without the use of ATP energy. So diffusion is a type of passive transport, but when we talk about simple diffusion, it's not going to be like, diffusion of a substance through a beaker of water. It's going to be diffusion of a substance across a membrane. So some substances, if you were to put like a solution of the molecules here that are orange are the solute, and then you have water as the solvent, right? So you have an aqueous solution of some molecule here of dye. If you have that a solution of dye on one side of a membrane, and the pores in the membrane are large enough to allow the dye molecules to pass, then they're just going to, by diffusion, they're just going to naturally follow their concentration gradient, which means concentration difference. There's a concentration gradient or difference on each side of this membrane that's being shown here. And this membrane is selectively permeable because it has these pores that are only going to allow molecules of a certain size to pass. So this molecule will move through those pores to the other side of that membrane until the solution on both sides is about the same concentration of that solute molecule. But the problem is, and you can watch that in this video, I wouldn't, it's animation slide 44, it's just on diffusion. But the problem is that in cells, the plasma membrane is selectively permeable. And this picture doesn't show that. This picture shows what would happen if the plasma membrane had these large pores in it, right? And it does not, but let's just pretend it did. All right. So in this case, you have a solution of orange dye on the left, a solution of purple dye on the right. What's going to happen over time is the orange dye is going to diffuse to the right side until the solution of orange dye is at equilibrium. And then the purple dye is going to diffuse to the left until the purple dye is in equilibrium. So you have a solution that's got orange and purple dye in equal concentrations. But if you have a membrane, I know if there's a picture coming up that will help you understand this better. Okay, so if you have a membrane and the pores of that membrane are little, then the solute molecule, in this case sugar, the green sugar molecules are too big to pass through those pores, but water can still move through. Water is a smaller molecule than sugar. So water can still pass through the pores. And the solutions just naturally are going to, I guess the action is going to be one that's going to try and make the concentrations equal on both sides of that membrane. So whatever happens is going to attempt to have an equal concentration of sugar on both sides. So if the sugar can't cross, where the sugar's concentration is high on the right side of this U-tube, and this is called a U-tube, by the way. It's a glass tube that we use in biology lab, to show movement across a selectively permeable membrane. So you put two different solutions on each side of the membrane, and then so the higher concentration of sugar on the right side, if the pores were large enough, the sugar would move to the left, and it would spread out until you had an equal concentration of sugar on each side. But since the pores are small, water is going to move toward the higher solute concentration. I always tell students to remember water follows solute. So water is going to move toward the higher solute concentration, and you see the water level lower in the left side and it rises in the right side. And you can also see that the concentration of sugar on each side is now a lot more evenly distributed. So it's reached equilibrium, not by sugar moving to the other side, but by water moving to the other side and basically diluting the right side that had the higher concentration. until both sides are in equilibrium with one another. So that's a type of passive transport, osmosis. So there's no, I'm sorry, y'all. My cat just got into my food. Well, since you just licked it, you might as well eat it because I don't want it now. So my cat's name is Shelby, by the way. And it's a boy cat. We named him after the Shelby Mustangs. So that's a little personal information there you may or may not care about. But anyway, the passive transport, which is transport that does not require energy. There's like different types of passive transport. I was going to see if they had a list. There's simple diffusion, and then there's osmosis, and then there's facilitated diffusion. So simple diffusion is what we talked about when I explained to you that some molecules could move directly through the phospholipid bilayer. So back here, okay, when I told you that molecules that are nonpolar, like lipids, fatty acids, which are part of the lipids, they're part of the building blocks of lipids, carbon dioxide and oxygen gas, all of these substances are insoluble in water, but they're very soluble in the phospholipid bilayer. So they can just simply diffuse through the phospholipid bilayer. That is called simple diffusion. Now, substances that need a transport protein are going to diffuse by facilitated transport. They're going to use a carrier protein to help them move from their high concentration to low. But osmosis is when water moves across the membrane. It's the diffusion of water because the membrane is not permeable to the solutes. So remember this with osmosis. Always remember water follows solute. Water is going to move toward, remember in our example, water moved toward the higher solute concentration. In this case, the higher sugar concentration. There's a video on osmosis. I do encourage you to watch this one. It's slide number 50. The word tonicity refers to basically the concentration of solutions that cells are placed into will determine whether the cell changes size, whether it shrinks or whether it swells or whether it just stays the same size. So tonicity refers to the concentration of solution that a cell is placed into. Isotonic means that the concentration of solute is the same as inside the cell. Hypertonic means the concentration of solute is greater than inside the cell. And hypotonic means the solute concentration is lower than inside the cell. So this is what happens to a red blood cell placed in a hypotonic solution. So what kind of solution would be hypotonic to the cytosol inside the red blood cell? Well, red blood cells have a salt concentration of 0.9%. So inside here is 0.9% salt, and what would it be? 99.1% water. I'm having to think about numbers in my head, and I'm not good at it. But anyway, so if we just place this red blood cell into distilled water that had no salt at all, distilled water is hypotonic to this red blood cell, okay? So what is water going to do? Water follows solute. In this case, the solute is the salt. Water moves where there's more salt. Since there's none outside, because it's distilled water, water is going to move into the cell and follow the little bit of salt that's inside, okay? By the way, that 0.9% is also in medical fluid, like in an IV fluid bag, is called normal saline. So if a person needs fluid, they give them a transfusion of normal saline solution, because if they gave them just distilled water, their red blood cells would do this. They would swell up, they would burst, and we call that lyse. When a cell bursts, we say it lyses. With a red blood cell, a lot of times they'll use a term hemolysis, because like red blood cells have hemoglobin. So sometimes this is called, when this happens, it's called hemolysis. Now, it's different in a plant cell. When a plant cell is placed into a hypotonic solution, it's a good thing. So if we place a plant cell into not a solution, but just into distilled water, right? So distilled water will be hypotonic to the fluid inside the plant cell. That's going to cause water to move into the plant cell, but it's not going to burst. It's going to fill this central vacuole, and it's going to cause the cell to build up pressure called turgor pressure. So we say the plant cell becomes turgid. This is the condition where we want our plant cells to be. This is what happens when we water our plants. Now, when we don't water our plants, they go flaccid, I think is how you say that. They go flaccid. That means they wilt in isotonic solution. So isotonic solution to a plant is not great. We want to water a plant not with an isotonic solution of normal saline, but we want to water it with distilled water or just water out of the tap that is hypotonic to the plant cell. If you place a red blood cell in an isotonic solution, what would be isotonic would be the 0.9% saline or salt. Then there's going to be some water that moves in and some water that moves out, but it'll be the same amount. So the cell will not change shape. It will not shrink. It will not burst. It will stay the same size. But in a plant cell, this is what happens when our plant wilts because we need to water it. Now, in a red blood cell that you place in salt water, and I mean like a higher percentage of salt, it can even be 1% salt would be saltier than what's inside the cell. So we place our red blood cell in a hypertonic solution of salt water. Then water's going to move toward that salt. It's going to follow solute. It's going to leave the cell. The cell is going to shrink or shrivel, and there's a name for that. It's called crenation. In a red blood cell. The red blood cell shrinks due to being placed in a hypertonic solution. We call it crenation. Now, a plant cell is going to die if it's placed in a hypertonic solution. If we try to water our plant with salt water, it's going to cause the plant to die. And what we see happening is the cell wall of the plant doesn't change, but the plasma membrane, see how it pulls away from the cell wall and the water leaves the cell through the plasma membrane, and it basically is just going to, we call this plasmolysis, and because it's losing cytoplasm, and it's going to shrink so much, it's going to lose so much water that it dies. All right, so in your notes, if you just want to make sure you know, isotonic solution has an equal concentration of solute than inside the cell. Hypertonic has a higher solute concentration than inside the cell. And hypotonic has a lower solute concentration than inside the cell. And I'm sorry, the other part is the volume of the cell, the shape of the cell will not change in an isotonic solution. So a cell without a cell wall is going to be stable in an isotonic solution. In a hypertonic solution, cells are going to lose water, shrink, and likely die. In a hypotonic solution, the cell will gain water and it will swell, and it could lyse or burst unless it's a plant cell, in which case it's just going to maintain turgor pressure. This page has an example about the paramecium that we saw on the very first slide of chapter 6. Paramecium are protozoans. They're unicellular. They have eukaryotic cells, so they have a nucleus and they have organelles, and they have a special organelle called a contractile vacuole. Paramecium is a type of protozoan. It's in the kingdom Protista, and it lives in fresh water, so it's constantly taking on water because the water is hypotonic compared to inside of the paramecium. So it has a contractile vacuole that pumps water out of the cell so that it won't swell up and burst. And this shows you the paramecium. I think paramecia look kind of like a little bedroom slipper, like one of the thin bedroom slippers. Anyway, you can see on the outside that it's covered in little hairs called cilia that help it to swim through the water. And it's got a contractile vacuole that's going to pump water out so it doesn't swell up and burst. There's a video, and you can watch this happen. And I think you should watch this video because I just think it's kind of cool to see protozoans, you know, under the microscope, living, alive, and moving around. That's slide 58. And this is going to show you the Elodea cells when they become turgid, when they're placed into a hypotonic environment, which they're already in a hypotonic environment because they're freshwater plants. So Elodea are freshwater plants, and they're very, very thin. Their leaves are very thin. So you can just take a leaf from an Elodea and put it on a microscope slide, and you can see all the cells clearly. I'll tell you something else. What you're looking at, these green structures, are the chloroplast in the Elodea. Some people have Elodea in their aquariums. It's like an aquatic plant that people put in their aquariums with their fish. So this is slide 61. This is a good one to watch too. So it shows you what plant cells do when placed in a hypotonic solution and how they become turgid. And then flaccid is what the plant cell becomes when it's placed in an isotonic solution. It will become limp. And in a hypertonic solution, it will shrink through a process called plasmolysis. And you can see that happen in this video, which is plasmolysis is slide 64. All right, now, another type of diffusion, but it involves membrane proteins, carrier or channel proteins, is called facilitated diffusion or sometimes facilitated transport. It is a type of passive transport because it does not involve energy, and it moves substances from their high concentration to their low concentration. We say that is down or with its concentration gradient when it moves from high to low. So in facilitated diffusion, a substance is going to move toward its lower concentration through a channel or carrier protein. And this animation on facilitated diffusion is slide 66. Here's a picture of a channel protein. Again, it's carrying out facilitated diffusion through the channel protein. This substance, this orange triangular shaped substance, is the solute, and it is moving from its high to its low concentration through that channel protein. So it doesn't require energy. It's a passive process. But some channels do have gates. Some ion channels have gates, and those gates keep the channel closed unless a chemical binds to the gate to open it, or there's a voltage change that opens that gate. So this, again, is showing you facilitated transport or facilitated diffusion, is showing you with a carrier protein and how that's different from with a channel protein. You can see that the concentration of this substance, these little blue molecules outside the cell, high concentration outside the cell, low concentration inside. So they're going to diffuse or move by passive transport toward their low concentration inside the cell. This time they're going to do it by chemically binding with a carrier protein, causing the carrier protein to change shape and open up and transport them inside the cell. So that is facilitated transport, but active transport is actually, this is confusing, but okay. Facilitated transport is a type of passive transport. Facilitated diffusion or facilitated transport is a passive transport because the solute is moving down its concentration gradient and no energy is required. But sometimes substances need to still keep moving toward their high concentration, and that's going to require energy because they're moving against their concentration gradient. So we call it active transport anytime a substance is moving against its concentration gradient. Not from high to low, but from low to high, toward its high concentration. Okay? So active transport always requires energy, usually in the form of ATP. and it will involve carrier proteins, which when they're used for active transport, a lot of times we call them pumps. So an example of active transport in cells is the sodium-potassium pump. It uses the energy of ATP to pump 3 sodium ions out of the cell, as you can see is happening here, and then 2 potassium ions into the cell. So sodium is moving against its concentration outside the cell where its concentration is already high. And potassium is moving into the cell where its concentration is already high. So the sodium-potassium pump builds up a high concentration of sodium in the extracellular fluid and a high concentration of potassium in the intracellular fluid inside the cell. And here's I would watch this animation. This is an animation on active transport, slide 77. And we're going to stop here. I will begin the next recording with slide 78.